And GBPNIHESD National Institute is an autonomous institute of Ministry of Environment, Forest and Climate Change, and its vision is ecological and economic security for sustainable development in Himalayan region. So the institute is working in the harmony with the sustainable development goal of United Nations, and the institute is working in several aspects, from water, socio-economic, biotechnology, biodiversity conservation, so many aspects, and one of the important aspects is medicinal plants. So why medicinal plant? Because as we know that there are several ingredients we can derive from the natural plants. Then they are having low production costs. They grow in wild, and raw material is available. They are more equitable and acceptable, and they are having less side effects as compared to the allopathic medicine, and better managed and affordable and source of livelihood. Next slide, please. I will be in little hurry because I have less time. Here in this slide, you can see the distribution, because our institute is working in entire Himalayan region, from Sikkim, Himachal Pradesh, Arunachal Pradesh, and Garhwal Regional Center, and Almora. Our institute is having the headquarters, and recently we have opened a new unit in Ladakh Regional Center in Leh. So entire Himalayan region is our area of working, and a total number of 22% plants obtained from the Indian Himalayan region, they are of medicinal value. Means then 1748 plants are of medicinal value. Next slide, please. And when we go through taxonomy, it is found that a total number of 223 family of the plants, they are having the medicinal importance. And among them, 191 is angiosperm, gymnosperm, and pteridophyte. You can see in this slide. I am not going in detail. Next slide, please. And when we talk about the medicinal plants, then medicinal plants is exports in different form, and medicinal plants are exported in the form of medicinal plants and crude drugs, herbal extract, concentrates, herbal medicine, Ayurvedic medicine, nutraceutical, cosmeceutical, dietary supplements, and health and functional food. Next slide, please. When we talk about the export of the medicinal plant, India is the leading country from 2011, and recently this is the statistics obtained from the Pharmaxazil, and that is the United Nations Commodity-Based Trade Statistics. And India is having the 19,000 million US dollar trade during the year of 2019, and this is the recent report. Next slide, please. These are the few prioritized species for Indian Himalayan region, and who is having the consumption of over 100 metric ton. Next slide, please. These are the prioritized species for Himalaya. I will not go through each and every one. Next slide, please. When we compare the demand and supply of the medicinal plant, and cultivation of the medicinal plant, then cultivation of the medicinal plant is far behind than the demand. So from where we are getting the medicinal plant for this export purpose, they all are obtained from the wild, and so there is a lot of pressure on their natural habitat, and there is need to conservation and sustainable utilization of these, and there is also need to cultivate these plants and to bring them into the cultivation, so that we can use recent innovation and advancement for breeding as well as for improving the variety of medicinal plants. Next slide, please. And here you can see in the slide that there is approximately 120 species are reported as threatened for this particular area. Next slide, please. And when we talk about the medicinal plants, then simultaneously we have to talk about the conservation. You all are aware about that, and I will deal with the access to conservation on which the GBPNIHESD Institute is working. Next slide, please. Now the activities of the GBPNIHESD Institute for medicinal plant promotion, cultivation and promotion. Here in this slide, you can see the institute has established a live gene bank and germplasm in 4 regional centers of the institute. One is Suryakund, where there we conserve 90 species. One is located in Himachal Pradesh, one is located in Garhwal unit, and one is located in Sikkim unit. And approximately 150 species has been planted in these herbal garden, and then we are maintaining the extension of these extension from different population and monitoring the growth of these medicinal plants. Next slide, please. We are also trying to propagate these plants by the seeds, and the protocol of approximately 25 medicinal plants of high value. For them, we standardize and establish the protocol. Here is the detail of the slide, and if anybody is required any detail, then later on I will share with you. Next slide, please. We have also attempted to propagate the medicinal plants by vegetative means, and in this way, GBPNIHESD has worked out for 15 species, and these are Podophyllum hexandrum and various other species. Next slide, please. You know, if any plant is, the availability of any plant is less in nature, then we have to propagate it through the tissue culture techniques. And we are also working on tissue culture techniques, and we have standardized the protocol for 20 medicinal plants. And then if you are having the propagated plants from tissue culture, then there is need to check the active constituents for which this particular plant is important. There is need to check that, and also there is need to check the variability of that plant, whether that plant is sumoculol variants or not. That is also needed to be checked. We are following the tissue culture protocol for standardizing until that GP Institute has worked out for over 20 species. We are also ensuring we are also promoting the cultivation of the medicinal plants by working with the farmers, and this is for ensuring the maintenance of the wild stock and to meet the increasing market demand. As you know, when you ask for a company to come in your field, then you should have quantum as well as the quality of the plant. So that should be ensured, so that the demand of pharmaceuticals industry should be fulfilled. If anybody wish to come in your area, then you should have sufficient quantum as well as the quality of the medicinal plant. Next slide. And this was a small attempt done by us. A total number of 5 medicinal plant species was cultivated, and they were planted in the Champawat district of Uttarakhand, and 32 family of the medicinal plant was distributed to the 32 family we distributed the medicinal plant in near Rudraprayag Sirsi village, and then we linked up these farmers with the traders, Imami and Dabur company. Next slide, please. Here, GBPNIHESD Institute has tried to remove the middleman, so that the farmer will get the benefit. In this way, we have some linkages with the HRDI Herbal Drug Research Institute that is located in Govind. So what we are doing, that we are working with the farmers. We are contacting with the person from the HRDI, and then we are signing their MOU, so that they will MOU with the traders like Imami and the person from Imami, the person from Dabur, so that they can get benefit. Next slide, please. These are few medicinal plants in the high altitude area of Indian Himalayan region, and we are working on these medicinal plants. I have mentioned only few plants. Next slide, please. Now, when we talk about the conservation, although the conservation of the medicinal plant is very essential, but we cannot conserve each and every plant and every population. So there is need to prioritize these plants and population, and by using some modern tools. And I have taken a case study so that for your better understanding. Next slide, please. In this slide, what we did that we collected the medicinal plant Valna Jatamasi from 25 distant locations, ranging from 1,000 altitude range to 2,700 meter ASL, and then we analyzed the morphology of these plants by using the morphological descriptor, which is given by NBPGR, National Bureau of Plant Genetic Resources, New Delhi. That is very authentic. Then we also analyzed the active constituent, because the value of a medicinal plant is live with the active constituent. If any pharma company will come to you and your plant is not of that much quality, then they will refuse to take your material. So we have to check first, before sending any plant for cultivation, we have to check its quality. And for that, in the case of Valeriana Jatamasi, valerianic acid and iridoids, that is very common, which is used for sedative property. So we have analyzed this, and then we analyzed our data, and it was found that as altitude increases, the quality and quantum of these active constituents is also increasing, and definitely there is some physiological role of the physiology of that plant as well as the environmental factor. Next slide, please. Similarly, we analyzed the data across the altitude habitat types, so we found that the pine habitat is best. This is a little bit lab kind of research. There are so many approaches to approve your results. If you are getting the high value for a medicinal plant, then it should have better DNA damage preventing activity, and that is a different approach. So we get a high value of medicinal plant in particular sample, then we have to authenticate it by using the DNA damage prevention activity assay, and in this way we approve that the plants, the Valeriana Jatamasi, the 100 microgram sample of Valeriana Jatamasi is able to protect the DNA of 50 microliter. Then there is need to analyze the genetic diversity for conservation and prioritization. We analyze the genetic diversity of the Valeriana Jatamasi across among the 25 population collected from 151 individual from 25 population, and we use 2 modern approach by using ISR and SSR marker. Next slide, please. And then we establish the relationship. Here you can see the DNA binding pattern, and these are related with for each sample. These 1 to 25 sample, they are representing each population. So next slide. This is just I'm trying to tell you that what kind of labor you have to do, what kind of work and what kind of research you have to do in this. Because I am a person from laboratory background, so I am trying to link my research lab work with the land. So once you are having the result, we analyze our data and we found that the genetic diversity of the species is decreasing with the increasing altitude, and the plants which are growing in open grassy slope where the sunlight is high, in these plants the diversity or genetic diversity is higher. So we hypothesize that why we are getting this kind of result. So we again recheck it by analyzing our data again, and then doing, because for maintaining the genetic diversity of a species, 1 minute more, sir, 1 more minute, sir. By analyzing, once you analyze for maintaining the genetic diversity of a species, you need to have the data on pollination, because cross pollination is play very important role for maintaining genetic diversity. So we analyze the genetic diversity of the species across the altitude as well as in the habitat, and we approve our hypothesis. So this kind of research we are doing in our GBPN, and these are the quite this is very well accepted study that is recently published in Pant, India, and this flora journal, that is very authoritative journal from life science. Next slide, please. So we are also trying to analyze the active constituents of various other medicinal plants like Rosia procera. So you are collecting the plants, so you cannot say that this plant is best source. Means that if you are collecting it at any season, so you cannot say. You have to analyze according to the season, according to the day, according to the month, and then you have to come out with the good result. Next slide, please. This is all about these are a few recommendations I would like to read. Prioritization of medicinal plants is very necessary. Then establishment of laboratory, high-tech necessary is required. Our adequate awareness program is needed. Use of modern science is also needed. Thank you. At last, I want to thank Director GBPN, Seva Foundation, and all participants.